Hello and welcome to this week's What Were They Thinking? Among the news items we have for you this week is a throwback to ancient history and the spread of various pathogenic organisms from rodents, the difficulty in developing a vaccine for malaria, Japan trying to drop a massive turbine into the ocean to start producing energy, NASA finding signs of life in space, and how one small rock in space has thrown a spanner in the works of the James Webb Telescope. The timestamps for these and other news items can be found in the description box below. Let's start with ancient history and a throwback to the spread of pathogens in the Dark Ages. Various rodents in Sweden have been found to carry a novel version of the coronavirus that we are all harboring concerns about now. In this case, we're talking about the red-backed bank vole, a relatively small creature looking somewhat like a cross between a rat and a mouse. The coronavirus they found within the voles is called the Grimso virus, primarily named after its location of discovery. It's not yet known if this particular variant of the coronavirus family is going to infect humans. However, based on past history, there is a possibility that the disease could spread via an intermediary, like fleas, to humans. This is exactly what happened with bubonic plague in the Dark Ages, something responsible for killing a very large part of Europe's population. While there's no immediate concern to be worried about this particular virus, it is known that the vole species in question has a habit of moving into human residences over winter when the weather turns cold. This will mean that come next winter, the risk of exposure will be significantly higher due to the proximity of the creatures and the increased probability of interactions. This is only further reinforced by the fact that the Grimso virus is particularly different to the other variants that are spread throughout Europe. There are a number of them, and they all come under the umbrella of coronaviruses. This just happens to be a very divergent variant and is indicative of greater adaptation to new hosts and habitats. In fact, minks were culled in the early part of the COVID-19 outbreak because the virus had spread from humans to farmed minks, and it completely destroyed the industry of mink fur, particularly in places like Sweden, Denmark, and Norway. While on the topic of COVID-19 and the question of creating a formulation of vaccines that aren't going to maintain nothing better than the status quo. Most people so far have had their 1 or 2 dose initial vaccination. Then they've had a series of boosters thereafter, anywhere between 1 and 3. These boosters are fundamentally exactly the same vaccine, just a slightly smaller quantity that ensures the immune system stays current and has continued memory of the virus in question. This means that there is a certain degree of waning efficacy. This is why there is a number of issues. The first and perhaps most obvious is that waning efficacy. New vaccines will need to be developed for new variants as time goes on. Exactly the same considerations that go into the annual influenza shots. The second consideration is that there is less and less willingness to get booster shots that are the same vaccine and don't deal with the virus. The initial impetus for people to take up vaccines was to let them go back to a relatively normal life with very few of the limitations that were in effect. This has obviously worked to some extent, and most populations have returned to a relative degree of normality. The challenge will make a vaccine that people need only have once a year and not get a booster, or preferably one vaccine that will provide much longer term protection than just a seasonal variant. And this is why the challenges are going to be, how do you create this? If we look at COVID-19 in the same way we look at influenza, it's both easier and harder to get that prediction of what particular variants will be a concern. Even with influenza, where we have a lot more data, it's not always right, as was the case in 2017. That is not to say these vaccines have no purpose. There is still a reduction in the severity and duration of infection because the immune system has some sort of priming towards being resistant to infection with those influenza strains, even if it's not a perfect match. This is one reason why influenza works so well. The difference between COVID-19 and influenza is that COVID-19 has a much narrower spread of variants at the moment, and so adopting a similar system of surveillance as for influenza might make it easier to create appropriate vaccines. These vaccines can take up to 2 years to prepare a large enough stockpile to be distributed and provide protection to the entire population. The trouble with that particular plan is not the idea. The idea is sound. It's the limited time frame to create that stockpile of vaccines. COVID-19 seems to have a fairly regular wave of between 4 and 7 months. At each time frame, a new variant emerges and becomes dominant. Influenza follows a somewhat different pattern. It tends to move from the northern hemisphere, where it stays for 6 months, to the southern hemisphere, where it stays for 6 months. This means you have nearly a full year where the same strains will move from north to south and then back again as mutations arise. The same time frame is not available with COVID, and so there's a much shorter lead up, and you need to have more predictive power. The best solution at this stage would be a universal vaccine, something like what we've past described with the Walter Reed Army Research Center, where they were developing a vaccine with up to 20 possible antigen sites that could be used in it, and that each particular carrier of these antigen sites could generate a much more systemic and complete coverage, even for as yet unoccurring versions of the COVID-19 virus. This means, in theory, you could provide protection for not only the known variants that are existing now and have existed so far, and you could also include future protection for the most likely variants to occur going forwards. Vaccines are the best example of why it is hard to do this at times, and none so much as the malaria vaccine. Malaria has been an endemic disease in many countries for a long time, and it is extremely difficult to deal with. It's a parasite that lives in red blood cells, procreates, breaks down the red blood cells, and goes on to infect more. It's transmitted via blood directly between two hosts, or via an intermediary like mosquitoes carrying blood between hosts. Because of how damaging it can be both to individuals and society, there has been a huge push to get a vaccine of some kind for it, but it's not been easy, and that's not for lack of money or trying. An article from the Smithsonian goes into some detail on both the pathway from developing the original primary ingredient in a promising vaccine for malaria, and some of the reasons they believe why it didn't come to market any earlier than it has. The original and primary ingredient used in a vaccine that reduces severe malaria symptoms by 30% was developed in 1990s. That is, it's almost a 35-year-old functional ingredient, but we haven't been able to put it into a vaccine in that time. Or it might be more accurate to say we haven't put it in a vaccine in that time rather than we couldn't. They attribute the primary reason that it's taken so long to apathy. The people who are most infected by this are located in Africa. Therefore, they're not Europeans, Americans, and so on. And they claim that this is the reason why there has been less drive to getting this out and working. Considering that between 14,000 and 51,000 children die each year based on the lack of vaccines and directly due to malaria, they believe that this is a very profound impact. There's a further 3 to 10 million malaria cases that could have been stopped by giving them vaccines like the one that's being trialled. Another reason was very similar to what we're seeing with COVID-19. The virus in COVID-19 changes regularly. With malaria, it goes through 3 major life cycles. Each time the life cycle changes, the particular site that would be effectively targeted is no longer available. And this is why the primary ingredient for that vaccine couldn't be deployed in a way that was going to assure effectiveness at all stages. You would have to get it at one point in time and try and eradicate the infection in its entirety at that moment. Then again, there's also considerations as to the changes in technology. Consider that we're using very novel RNA vaccine technology for some of the most effective vaccines used for COVID-19. Things like that did not exist when they were trying to develop these vaccines for malaria. This research did not come out of nowhere, and like a lot of things that are found in the biomedical research area, a lot of funding comes from the US military, and that's one of the reasons they found what they did. The US Army was trying to fund a vaccine for soldiers deployed overseas, malaria being something that could put them out of action for a very long time. The author of this article claims that outside of that particular niche area of interest, everything else was academic, and they were interested in the research element, not the public health element. There were also the logistical and practical considerations of trying to run clinical trials in Africa. They fundamentally had to set up all the infrastructure for creating the vaccines, running their clinical trials, and trying to figure out what was working all on site and from nothing. A huge logistical hurdle that took a long time and a lot of money to overcome. In fact, it took 20 years from the first clinical trials being set up and run to the eventual deployment of a facility that could produce the vaccine and then start issuing it. This is on top of the prior decades of research into finding what was needed for the vaccine. The article then goes on to try and make some parallels between COVID-19 and malaria and the lack of interest, funding, and effort. Unfortunately, these people seem to have missed some of the more important factors. One is that most of the vaccine research funding came from governments, and those governments are not located in Africa. If the argument could be made that African governments made the same push, then you could say quite reasonably that yes, they were dealing with something that they had to deal with for their own public health benefits. The same way places like America, most of Europe, Australia, Canada, and so on, all pushed money into research on the basis that it was for their public health benefit. Malaria is not a problem for them. They still provide funding and research, but it is not as much of a priority for them. The same sort of issues apply to not just research focusing on diseases that are able to be communicated, but diseases that occur outside of that. And generally we're looking at fundamental or basic research. That is, what is going on in this particular thing? Can we describe it, tell you what it is, and eventually somebody else can explain how it works from what is being established. Foundational research is necessary for everything else that comes later. Even creation of vaccines relies heavily on foundational research. This is why the description of differences in neurons between primates and non-primates, for instance, is a very compelling bit of basic or foundational research. Structural differences and very clear structural differences exist between primates and non-primates in neurons. This could be very important if we are, for example, developing vaccines that rely on looking at changes in the brain structure from, say, a mouse model and converting that into a human model. You can't always rely on it, and mouse models have been notoriously known for their difficulty in converting from research in the rodent model to the human model. One of the first notable differences was that dendrites, which are small projections that help to receive signals from other neurons and grow out of the cell body, were thought not to have any axons, or the extension from the nerve body that carries the signal to the end where it releases the signal to be picked up by another neuron, or where it's meant to have its action. In the case of non-primates, it would appear that the dendrites, which shouldn't have axons in primates, have them much more frequently, and this could change our fundamental understanding of how the neurons interact in the brains of non-primates. This is the kind of research that could have big ramifications for other research into more conventional diseases like Alzheimer's. It goes from that sort of foundational research to clinically relevant research, and in this case, how the proteins involved in Alzheimer's can cause increases in heat in the brain, causing the neurons to become damaged and therefore begin to fail if not die off entirely. The protein bundles or tau bundles in the brain during Alzheimer's are one of the bigger challenges. They don't go away, and so understanding not only how they're generated, but what they're doing once they are generated will be key to trying to solve them, and then solve Alzheimer's as a result. The bundles of protein in the brain appear to be causing overheating, and that overheating is then leading to those neurons becoming damaged. This leads to cell death and the brain begins to shrink. This then leads to the complications that we see in those with Alzheimer's disease: decreased neurological function, tics, spasms, and more. Not only is the increase in heat going to cause damage to the cells, particularly the neurons, it could also increase the rate at which the tau amyloid bundles begin to aggregate, which would only serve to exacerbate the signs, symptoms, and effects of Alzheimer's. This would be effectively an ongoing positive feedback loop, where increased heat causes more symptoms and signs and eventual death, but along the way it increases the cause, the tau bundles, that lead to that effect. Going from clinical and medical news to technology and the creation and deployment of a huge turbine. Wave generation is not a new thing. In fact, using the waves beneath the ocean is something that has been tried for a very long time. There are a number of companies that have tried to create and then sell this technology, some more successfully than others. Most of them have been sent off to areas with relatively limited access to resources. For example, isolated islands, where the wave generator not only provides movement of water, desalination, but also energy. Japan's attempt is to put this huge system into the ocean, and the movement of the waves up and down under the water will allow for the creation of electricity. The generator is about 20 meters across, or roughly 60 feet, the same in length, and about 11 meters, or approximately 33 feet in height. The area they're looking to deploy this system, and possibly more in the future, is called the North Pacific Gyre. It's a relatively smallish area where they figure they can get up to 205 gigawatts of electricity, or the roughly current equivalent to their national production of energy. The advantages of this design over the comparatively more effective wind turbines that produce 3.6 megawatts is that by sitting roughly 50 meters below the surface, it's able to avoid any of the issues to do with typhoons and similar. It also gets a relatively constant movement of water through it of between 1 to 2 meters per second, or 2 to 4 knots. This produces significantly less power at 100 kilowatts versus 3.6 megawatts, but it's also much more resistant to any of the environmental factors that could hit it. The model they're looking to deploy now, the rather large 20 by 20 by 11 meter system, could produce 2 megawatts, which is much closer to what offshore wind turbines could make, but it's also not yet deployed, so the exact production numbers aren't clear yet. Other technology news builds on the issues that face wave generation, and that is, small technical failures can lead to the entire project going under. This is somewhat what the James Webb Space Telescope is currently facing. It's been hit by a small rock, and this has had a big impact on its ability to work at the moment. Given that the telescope was deployed 1.5 million kilometers, or about 1 million miles from Earth, the entire system was built with redundancy and a certain buffer within its operational ability. The functionality is not going to be affected once they've adapted to what has been put out of action. And this is where we need to look at just how they've done this. One of the things they've done is that the entire satellite was built with the ability to not only detect what small dust fragments would be knocking individual mirror sections out of alignment, but to then detect what that misalignment is and reposition the mirror accordingly. This would then theoretically fix for any distortion that would occur. If there's going to be larger things that are likely to hit the telescope, they also have the ability to rotate the telescope away from it so that it's not facing towards them and would hopefully not be affected, or if it is, affected significantly less. Other astronomy-related news for this week is the approximate alignment of Mercury, Venus, Mars, Jupiter, and Saturn along the elliptic plane. You should be able to see them sometime near dawn for the entirety of this month. This is the first time in about 18 years that this alignment has occurred. Depending on where you live, your chances of being able to see all 5 planets are going to vary considerably. For the most part, this will be those in America and the Northern Hemisphere as a whole. You also may not be able to see Mercury very well, given that it will be very close to the horizon and not as obvious as the other 4 planets. Our final item for this week is the discovery of the early building blocks of life on an asteroid. It's taken quite some time now for the Japanese Space Agency to bring back its samples from Ryugu, a meteorite or asteroid circling in space. What they found are 20 amino acids on this chunk of rock floating in space. A chunk of rock more than 320 million kilometers from Earth, or 200 miles from Earth. It is a very clear sign that early building blocks for life, like amino acids, can occur and develop in incredibly inhospitable environments. Given how old Ryugu is and the fact that it's come into our solar system, would indicate that it is a very strong contender for the argument that the origins of life for Earth are not terrestrial. It further reiterates the possibility that asteroids or meteorites, as they've gone through the solar system and the rest of space for that matter, could have been seeding planets with the necessary biological material to give rise to life, if the planet was hospitable enough. That's all the news we have for you this week. Thank you for watching. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.